So good morning and welcome to the first of the Spring 2024 Institute for Quantitative Biomedicine seminars. I'm Stephen Burley, the founding director of the Institute, and I'm very pleased that we're able to host a team, not just one, but two speakers from OpenEye Cadence Molecular Sciences today. Before I introduce our speakers, I did want to just give a shout out to the rest of the program, which will resume on the 14th of February. Remember that we don't clash with the CABM seminars that are the first Wednesday of every month. So I want to extend congratulations again. I think she's in the room this time to Erica McCarthy, who is the recipient of the 2024 IQB Inclusive Leadership Award in recognition of her outstanding achievements in training and mentorship. We are always looking out for individuals to join our IQB Diversity, Equity, and Inclusion Committee. Please ask Michelle for details if you're interested in joining. I want to remind you that there are a lot of opportunities here at rcsb.org for software developers, for recent graduates doing gap years, and for undergraduates doing summer internships. So please go to our jobs page, www.rcsb.org/jobs/pages/jobs for more information. And you'll see that there's a host of opportunities that we're currently recruiting for. I also, this is a particular shout out to depositors, but also to users of rcsb.org. That we have instituted a series of virtual office hours, and this starts on the 5th of February with Rachel Kramer-Green, who's a PhD graduate of Rutgers, and she is the scientific support and customer service lead for the RCSB Protein Data Bank. And she'll be hosting the first of these office hours on the 5th of February. You do need to register to get the link to join the the Zoom call. So if you're interested, please do so. It'll run from 2 to 3 p.m. Eastern time on the 5th of Feb. And I also want to draw your attention to the fact that in the following week, we are going to be offering an IQB crash course, a webinar that's put on by RCSB PDB, focused on visualizing biomolecular structures with MolStar and allowing you to go literally from looking at individual atoms of proteins, nucleic acids, etc., to being able to make movies of these structures from the PDB or computed structure models coming from the AlphaFold DB or the Model Archive. And this is scheduled for February the 13th. I think that's a Tuesday at 1 p.m. Eastern. And participation in this, like all of our IQB crash courses, participation is free, but registration is required. So please go to iqb.rutgers.edu or use the QR code on the screen to be able to connect to the registration page. So with that, I think I just want to acknowledge that IQB is a team. Science is a team sport, and running the infrastructure to support science is also a team sport, and to recognize the contributors to the Institute for Quantitative Biomedicine who are depicted on this last slide of mine. So with that, let me enter. If we could go one more, sorry, one. Yep. With that, I'll introduce now our two speakers. This is a tag team match. Leading off is Dr. Addison Smith from OpenEye Cadence Molecular Systems. He's a scientific software developer 2 in that organization, and a PhD in chemical engineering graduate from Brigham Young University. The title of his presentation is Protein Preparation in the Cloud and Expanding MMDS to All Protein Targets in the Protein Data Bank. And then he will be followed by David Weich, who has also a scientific developer too at OpenEye, Cadence Molecular Sciences. He has a PhD in pharmacological sciences from the University of California, Irvine. And previously he received a BA in physics from Claremont McKenna College, which I think is in California, right? Yep. Okay. So gentlemen, we're delighted to be hosting you. Thank you very much for agreeing to come, and our thanks also to Ashley Ball for joining them, and the productive conversations that we've had this morning about strengthening the ties between the RCSB Protein Data Bank and OpenEye. So thank you.
Thank you. Okay. I'm just going to pull up my presentation. Let me actually kill the share. Okay, sweet. Thank you all for letting me have this opportunity to come and talk. As was introduced, I'll be talking about protein preparation and talking about how we do this in the cloud on a very massive scale. So just a quick introduction to my team, the team that I work on. It's led by Jasper. He's leads us in the biomodeling group, and we kind of have two sides to the group. We have scientific developers, and we also have GUI app developers. And so kind of just a brief introduction, and then introduction to me, just real quick. I grew up in Oregon, then made my way to Utah for some college, and then now I've made my way to Santa Fe, and it has been a wonderful journey. I am a twin, and identical twin, so maybe you can guess which one is the identical twin in the lower left-hand corner. And then this is my family. I got two little girls, and they keep me very busy. But now to the science, because that's what we're all here for. Okay, so just a quick question. Who here does like in silico work? I'm kind of curious. A lot of people, few people. And then the rest of you do wet lab biology stuff? Yes, no, maybe? Awesome. So just trying to get a figure of what everyone's background is, because what we kind of do in the biomodeling group, or at least one aspect of what we do, is kind of merging what we get from experimentalists, like from the PDB, and having those structures, and then moving it into an in silico environment where we would run MD and a bunch of other simulations. So we kind of work in kind of that mid space between the two, and we have to do oftentimes some of the structures we receive. They need to be modified in some ways. They need to be protonated. And then, so we use a tool called Spruce. It's our way to spruce up the protein. And here we have a kind of a workflow that we do. Because oftentimes experimentalists give us the exact coordinates, and sometimes loops are missing. Sometimes places don't have proper protonation states. And so we need to resolve some of these conflicts so that when we generate data from there, we have something that people can design on. And so that's why we call it a design unit, is because then some drug discovery scientists, they can use these design units to then design small molecule ligands with it. And then down here, just kind of some tools that we have internally that require design units. And yeah. Okay, so Iridium is another metric that we use to judge design units. So just because we have them prepared does not mean that they are perfect. And so we wanted a non-relative schema for determining if this is a trustworthy, and to judge the trustworthiness of the structures. And so this was developed by Greg Warren. There's a few people out in the audience that I think know who he is, and fun story about how he came up with that. Won't go over it here, but basically we have these kind of criteria that need to be hit in order for us to really trust something, and some of these are pretty strict. criteria. And if we can land and bin a structure into every single one of these, then we know that this is a very trustworthy structure and we can base our models off it. And then our docking algorithm obviously was trained on these criteria. So yes. So now we move on to Orion. Orion is our cloud platform where we can run lots of computations in the cloud, and it's primarily based on our toolkits, which is which is where a lot of our science is run from. And we run our calculations in things called flows. And so cubes and flows. Cubes are these little squares, cube things, and then the flow is the entire process. And so, excuse me, what the cloud gives us the benefit is parallelization. And so if we parallelize some of these processes, we can expand out from just one CPU, we can expand up to thousands of CPUs. And so then we can run computations quickly, and we can also get results on a very big scale. These flows are not as simple as that simple one at the bottom. Some of them can get very, very complicated. This was one that I developed for MMDS, and there's a lot of different things, and we will get into MMDS right now. We also take data from a bunch of different third party, and especially the PDB. We love the PDB because they provide great data for us. And then Orion platform kind of just marries all together. Okay, so MMDS. This is something that we have tried to develop to really visualize and bring these design tools and everything that I have just talked about onto a place where people can do drug discovery and view it in an environment that you can see all the different family relationships. So it's built, the public data is built off of all these public data that are represented here, PDB, Guide to Pharmacology, Uniprotin, they kind of all siphon into this MMDS structure that we have, and I will go into more detail. So this is kind of, there's a couple ways we can like interact and interface with MMDS, and the first way is to go through different project trees. And so we can see Guide to Pharmacology is just what I have right here, and it's showing all the different types of targets you could shoot for when you're doing drug discovery. And so you can break it down, and then enzymes will break down into further subcategories, and it's just a big long tree. And ultimately, at the end, you have a bunch of PDB codes and design units that are superposed on top of each other. You can also search for the exact PDB code, but they're superposed on top of each other, and we can then view the actual binding site that is we are interested in when doing it. So it's just comparing the two again. So like if we do a search in our CSB, we get a bunch of different structures. What we try to do is we take those structures and we kind of put them in families, and then we can then superpose them, and we can view what's there. And I know it's like taboo to do a live demonstration, but I'm going to try it. Sweet, it worked. Okay, so here we have the same structure. This is pulled up in MMDS, and we can see that there's a ligand inside of there, and then this blue structure is also APO. So we just have one, but we can then compare it to another structure, and then we can add all these structures, and we can see that we can add them together, and we can see where the functional parts of the binding pocket are. So clearly there's interactions here that is important for this binding site. And one of the structures kind of has clashes, so we can get rid of that. We can also view the surface. We can see, kind of get these tools together, and we can figure out how and what is important in the different contributions of the different groups. We have also tied this with AlphaFold. So if there is an AlphaFold structure that appropriately matches these design units, they will also show up. And I actually think that first one is an AlphaFold structure. So yeah, that was lucky. Okay, we'll go back. Okay. Yeah, this is in case my live demonstration didn't work. Okay, so kind of the organization part, we'll kind of say how, kind of go into how that works. There were a few challenges because oftentimes we don't have a nice target tree like guide to pharmacology, and then the PDB will just have these structures that aren't really hung on any sort of branch. And so we went into Uniprot, which had its own challenges, mostly due to the fact that you can have similar groups of proteins and like protein systems with different subtargets inside of it. But oftentimes a PDB will have multiple subproteins inside of it. So we had to break those apart. And that was the main mess that was inside of that first flow that I showed you guys. But yeah, this was definitely something that took some time to figure out how to parse them out and to identify these different types of proteins. Okay, are we not moving? There we go. Okay, so this is just a general status of what's in the MMDS. In total, there were 106 experiments from the PDB that we prepared, which is about half of what's actually in the PDB. We generated over 200,000 design units, and then it's organized into a couple different trees. Guide to pharmacology, there's just an uncategorized tree. We were able to include AlphaFold structures as long as there was another associated experimental structure with it. There was actually fewer than I was expecting. And we this has also led us to help us mine the data, which has led to improvements in our toolkits. And here's a few stats based on Iridium. So of the structures, about 16%, sorry, 20% of the PDB codes generated good design units, so that was promising. And we've actually been tracking this. This number has actually gotten much better over the years. So it was like 10 or 15 years ago, much lower than 20%. So we're very happy to see that the quality of data being inputted in the PDB is getting better. We're very happy about that. Okay, I alluded to AlphaFold. So this is an example where we have two AlphaFold structures. We have a ligand right here, and then we have what looks like a ligand coming in here, but if we zoom out, it's docking into itself. So AlphaFold definitely has some limitations, but we think it's a fantastic technology moving forward. Here's just a summary of some of those. As this ML and AI hype gets better, we hope to more fully incorporate it into our toolkits. It definitely has some very important uses in our field, and we're trying to hopefully filter out some of these weird cases and utilize it for as best possible. We are optimistic but realistic in its implementation. Okay, just a few more acknowledgements, and that ends my bit of the presentation. Thank you.